Okay, so when we're talking about substances getting in and out of a cell, we can categorize them by energy. Either it uses energy or it doesn't. If it doesn't use energy, we call it passive. Passive transport means substances can go in and out. No energy is required. Okay, so the first one we looked at was just simple diffusion. Okay, so diffusion is just the tendency for molecules to spread out. With or without a membrane, their tendency is to spread out evenly. If you put a drop of food coloring in a glass of water, you don't have to stir it. It's faster if you do, but if you just leave it for a little bit and come back, the food coloring automatically disperses evenly. That's diffusion. And so substances will do that also by going across a membrane, assuming they can get across. So since the membrane is made of 2 layers of phospholipids, we've looked at that before, the phosphate heads are polar and they face the outside and the inside of the cell. And they're much smaller than the fatty acid tails. Each phospholipid has 2 fatty acid tails, which are really long. Okay, so they face each other. That means the majority of the thickness of the membrane is nonpolar. So any substances that go right through the membrane have to be nonpolar, plus they have to be small, because they have to be smaller than the phospholipids in order to go through. So those substances, like carbon dioxide, can go through by simple diffusion. They'll go right through the membrane. Other substances that can't go through the membrane. Okay, so they're small, but they're polar. So polar things aren't going to be able to get through. They're going to have to find some other way. Okay, so they will find typically a protein, one of those transport proteins. In this case, however, since we're still talking about diffusion, it's still passive, this is a protein that is going to be, say, open down the center. Some of them are completely non-selective, where they're open, anything that's small and polar can go through, in or out. Other proteins are more selective, where they're only going to allow certain substances in or out. So if that's the case, and still no energy is involved, we call that facilitated diffusion. With facilitated diffusion, it's still diffusion, so it's still passive, but there's a protein involved. It's for small polar substances. In either case, these molecules are diffusing from higher concentration to lower concentration. Because we go from high to low, they call it going down the concentration gradient. It's like riding a bike downhill. It doesn't take any energy. It will continue until those things are equal on both sides. Once they reach that, it's called equilibrium. At that point, substances still move, but it's equal now in both directions. Whereas before, if you have more on one side, less on the other, they will diffuse mostly in the direction where there's fewer of them until it reaches equilibrium. Now here they're showing that cube of sugar. Same thing, plop it in some water. If you walk away and come back later, the whole thing will dissolve by itself, assuming there's enough water to dissolve all of it. That's because of diffusion. Osmosis is the diffusion of water across the membrane. Osmosis, that's the diffusion of water across the membrane. So it's only for water. That's our third passive transport. Okay, so we have diffusion, facilitated diffusion, and osmosis. There are special proteins special channels in the plasma membrane called aquaporins. Those are for water only. Because, especially for multicellular organisms, it's very important to be able to share the water from one cell to another and do it quickly. There are lots of these aquaporins in these cells, in the cell membranes, where water can go in and out. Osmosis occurs not just to share water, but also to keep the concentration wherever it's supposed to be. So for us, the concentration of salt is supposed to be 0.9% sodium chloride. That's saline solution. So you'll find that labeled on nasal sprays or eye drops or in typical saline in IV bags. Now you go in, if they want to give you some fluids, they're going to put you on a saline drip IV. Then if they want to give you extra stuff, whether it be medications, antibiotics, glucose, protein infusion, whatever it might be, they'll add that. But saline is just the typical 0.9% fluid that we find in our blood plasma, in our tissue fluids, and inside our cells. For plants, it's greater than 0.9%. It's saltier inside the cell. That keeps water coming in. Okay, where at 0.9%, where it's equal on both sides, that's equilibrium. And so water goes both directions, mostly equal. Now it's constantly moving either in or out based on what your body's doing. So with every second goes by, there's all kinds of chemical reactions occurring inside, outside the cells, some of which inside produces carbon dioxide. So that's raising inside the cell. It's going to be going out. Oxygen's coming in. Wastes are building up. They're going out. Nutrients are coming in. So there's constantly this back and forth motion, and water chases those things back and forth to try and keep all of the concentrations the same inside and outside the cell. It does that through these aquaporins. And there are some substances that attract water more than others. For example, sodium and chlorine, potassium. And a lot of ions have much stronger pull on that water than others. It can get pretty confusing, pretty tedious to try and figure out which way is water going to go because of so many of the things that we have on either side of that membrane. And so in general, the first step in talking about this stuff is just total numbers. We could say, okay, what's the total numbers of substances that are dissolved inside versus outside? Wherever it's higher, water will go in that direction. That's not entirely the truth, because if we start to look at what the various substances are, water is more attracted to sodium, for example, than it is a lot of other things. But for this class, in general, we're just going to say wherever there's more stuff, water goes there. And by osmosis, doesn't cost the cell any energy. Yeah, we saw that. All right, so talking about osmosis then, when we compare two solutions, so this is what we did last week in lab, when we compare two solutions, in this case, inside the cell versus outside the cell, the concentrations are either the same or they are not. If they are the same, we call that isotonic. Iso means same. So for us, that's 0.9% saline, inside versus outside. For most other mammals, it's also 0.9%. For any other organisms, they will have possibly their own percentage that is typical for inside versus outside the cell. For single cellular organisms, depends on where they live, whether they are truly isotonic to their surroundings or not. And for the little single-celled protists that we saw in pond water, probably not, because it's fresh water outside and they're going to be saltier inside, so that's why they had to have that, help me out, what vacuole was that? Contractile vacuole that helps to keep the water from bursting the cell, so they keep having to push the water back out. The water keeps coming in because it's saltier inside the cell. So under those conditions, so when it's isotonic, water moves, but it moves equally in both directions. So the cell's not gaining or losing water. If the concentration is different, outside versus inside, well then we can label the two solutions either hypertonic or hypotonic. The hypertonic solution is the one that has greater concentration. More molecules on the hypertonic side. The hypotonic solution has lower concentration, so there's fewer molecules. Which way does water go? Water always flows toward the hypertonic side. And keep that in your mind as a rule, so that if I tell you which way water is going, you should be able to tell me which side is hypertonic and which side is hypotonic. If I give you an example of some side that is hypertonic, then you know the other one's hypotonic. You'll know that water is going to go from the hypo to the hyper. And vice versa. Now, there's a couple of things that will hinder that. For one thing, the pressure. So if water's flowing toward the hypertonic side, and then the pressure starts to get really great in there, then that may put a stop to osmosis, or it flows to the hypertonic side until conditions become isotonic. The pressure that I was talking about, so the movement of water, if the water's moving mostly in one direction, think about standing in a stream. Not every water molecule is moving that direction, but most of them are. You can feel the pressure on your legs. So it's the same thing here, but on a much smaller scale. If we have water that's mostly moving either in or out, it creates a pressure gradient called osmotic pressure. So for example, in any capillary bed in your body, so you have arteries and arterioles that branch out, there's no exchange outside versus inside the arteries and arterioles. So you can't drop anything off or gain anything there. That has to be done at capillary beds. A capillary is a teeny tiny little vessel that is one cell layer thick around. So really, really thin walled. And when it gets there, you have one arteriole that'll lead in, and then all of this branching takes place, so that you get this whole capillary bed. Well, there's a difference in osmotic pressure at the beginning versus the end. So at the beginning of the capillary bed, that's mostly where substances will be dropped off. So all the nutrients, the oxygen, they're net going out, which means the osmotic pressure is going out. At the same time, there's going to be a buildup of wastes in that area from all the cells that needs to be picked up. It can't go against the osmotic pressure, so that usually happens on the back end. So the net pressure is outgoing in to the capillary bed, but then the net pressure is in at the end of the capillary bed. And then it all comes back together and goes back into venous return. So if your osmotic pressure is off, then that can be a big deal too, because that's going to mess with those capillary beds and nutrient drop off and waste pickup, that type of thing. This is from lab last week, but in animal cells, typically we need isotonic conditions. So their example here is just showing a red blood cell. And it's not just red blood cells that are affected by this. There's a lot of others too. So this is just one cell that's an example. They're supposed to look like this. It's called a biconcave disc, so that on either side, they're dented in the middle. Looks like a breath saver, a red breath saver. Notice there's no nucleus. Red blood cells don't have a nucleus, which means they don't have that full set of chromosomes and DNA because they don't divide. These are made in your bone marrow, and once they're made and released, their job is just to circulate around. They pick up oxygen at the lungs. They drop off carbon dioxide there. They do the opposite when they get to the capillary beds. So there's no reason for them to divide. Their chemical activities are pretty minimal. So the DNA, remember, is used for what? Making proteins. They don't have the DNA, so they're not going to really be making a lot of proteins. So their lifespan then is pretty short. I want to say it's like 40 days or something like that. So once these are made, they circulate for about 30 or 40 days or so, and then your spleen is responsible for taking them apart and cleaning your blood out of the worn out red blood cells, while your bone marrow will constantly produce more. Okay, so this is isotonic, 0.9%. Here, they're all shriveled up. Which way was water moving then? Out. Why? Why would water move out of the cell? What's out there? Which way does water always flow? Towards the hypertonic side. So we may not know exactly what's outside, but the concentration outside the cells in this picture is higher. So water's moving out. It could be various salts, it could be different ions, it could be proteins, who knows, but the concentration out here is higher, so the cells lose water. They don't function very well when they're like this. So that's not good for whatever mammal or animal we might be talking about, because the oxygen level is going to be lower. It can't carry oxygen like this. On the other side here, it's showing that water is moving in. Why would it do that? Where's the hypertonic side now? Inside. Okay, so whatever made that happen, it could be, in this case, that outside is watered down. So then water starts to move in. If it doesn't stop, if it does not reach equilibrium soon enough, then we'll see what's happening to this one. That red blood cell will burst. And as I pointed out in lab, this is a very nice, gentle picture of it. It kind of looks like a balloon. Really, it's like, firing a huge bomb at a balloon where it's going to be obliterated. There are so many small pieces, you won't be able to find any of them. And so that's what happens usually when these blow up. If it does reach equilibrium before it bursts, that's good. Then the conditions could be reversed given time or medical attention. So what would you do? What do you think happens if somebody goes into the ER and their red blood cells would look like this? How could they get water back out of them? What's the concentration percentage supposed to be? 0.9%. So it's moving in these cells initially because out here must be less than 0.9%. And water's tendency is to try and get the percentages the same. So whatever's going on here is less than 0.9%. So what could we do? So, yeah, give them some salt. So hook them up to an IV. Depending upon the extent of how much swelling has gone on, one thing is that they could just give that person 0.9%. And, the kidneys will work and flush out a lot of the excess water. And so they'll pee a lot, but within hours, their concentration would be back to 0.9% and their cells could go back to being normal. Will some of them be damaged? Probably, but thankfully the body has ways of healing that. In this case, we could do the same. And now over here, if it's too extreme and they want faster results, they won't give 0.9%. They might give 1% or something like that. Because the higher that goes, the faster that water is going to come out of these cells and return to normal. Here, we want to do the opposite. The percentage outside these cells has gone up, and so they're losing water. And this could happen, say, if somebody's really, really dehydrated. This may happen. So again, they'll have to assess how bad it is, but just hooking somebody up to regular IV fluids, 0.9%, will help to fix that. Down here, this is plant cells. Okay, so plant cells function better when they are immersed in a hypotonic solution. So it's hypertonic inside. That's why a lot of fruits and vegetables have either a very sugary taste, kind of sweet, or maybe even kind of salty. Now, what we taste is pretty minimal in terms, usually if you're talking about greens, in terms of, you know, sweetness or saltiness. But if you think about celery or some lettuces or sugar snap peas, there is a slight either saltiness or sweetness. That's because those plant cells have to maintain something that's greater than 0.9% inside to keep the water coming in. That creates a lot of pressure on the inside called turgor pressure. We call that cell turgid, and that's when the plant is nice and healthy and standing upright. If it starts to lose water, either in natural conditions because of a drought or a really hot, dry day, or if we're talking about house plants, you forget to water them for a while, then the cell becomes flaccid, where the pressure is equal on both sides. That's good for animal cells, not good for plant cells. So this is when it starts to wilt. If it continues to lose water, then we start seeing something like this, where the plasma membrane starts to pull off of the cell wall. This is called plasmolysis. So if you don't remember that from your lab, write it down again, because I'm sure it's on the test. Plasmolysis, that's shriveling of a plant cell. Shriveling of a plant cell. You should also know turgid and flaccid. So flaccid, turgid, make sure you know those. For animal cells, you remember what this is called? What's shriveling of a blood cell? Good. Crenation. Shriveling of an animal cell, typically it's specific to a blood cell. What's it called when it bursts? Good. Hemolysis. Cell lysis is bursting of any cell. Chemo here indicates that it's a blood cell. So bursting of a blood cell. Questions over any of that? So all of this is caused by osmosis. So no energy is expended for any of that. I'm going to come back to this page because I want to go over active transport before I get here. All right, selective permeability. You guys already know about this from lab, but it just means that the cell is selective about what it allows in and out and how fast. Some of it is passive that we've already talked about. But the next types we're going to talk about are active. If it's not passive, then it's active. Active transport, this involves protein pumps. Protein pumps, because if you look what it says right here, it's going against the concentration gradient, or you could say going up a concentration gradient. So because of that, these pumps are on trying to pump some substance all to one side. Well, the natural tendency for that substance, whatever it is to spread out and try to get back across the membrane until it's even. So these pumps in the cell membrane are constantly on, constantly pumping. Similar to a bilge pump on a boat that pumps out the underneath where water might accumulate. You turn it on, it pumps the water out. The difference here is these are always on. This is not active, so I already went over those. Selective and facilitated, that kind of goes together. Okay, so here, let's look at it. Here's what it might look like. Okay, so here's one protein. This one happens to be the sodium-potassium pump. They chose this because very, very important in our bodies, very important in multiple types of cells. Okay, so what's happening here is that sodium, ideally, is supposed to be pumped to the outside. Potassium is supposed to be pumped to the inside, but both of them have a tendency to be able to sneak across. So it never gets to be 100% on either side. Partly because they're sneaking in and out. The other part is because of us eating and breathing and doing all the things that we do, so it changes the concentrations. But these pumps then, there's many of them in the cell membrane. When it's open to the inside, it has 3 spots for sodium, so it can pick up 3. Notice this ATP molecule hanging around. Okay, so later we'll spend a whole chapter talking about ATP, but that's the energy that the cell needs to do things like this. So once it's loaded with the sodium, the ATP molecule transfers this last phosphate on right there. And so that's what we're seeing here. Here's the rest of the ATP. It's now called ADP for diphosphate instead of triphosphate. But when that phosphate joins, it transfers the energy and it changes the shape of that protein so that now it's open to the outside. These 3 sodiums will be kicked out, and for efficiency, 2 potassiums can be picked up while it's open to the outside, and they're going to fit right in here on the other side. Because of that efficiency where it can pump both directions, that costs the cell half the energy than it would have to spend if it had separate pumps. So that's good. Once that happens, the phosphate breaks back off, and this goes back to its normal shape and kicks those potassiums inside. And so it's constantly pumping like that. Every time it pumps, costs 1 ATP. If somebody is, brain dead in the hospital, do you have to feed them? Is their heart still pumping? Yep. So there's still some activity. So, yep, they'll feed them through IV, glucose solution. But yes, you still have to feed them. This is one of the reasons why. So they're not actively getting up, walking around. There's not a lot of brain activity that's using up a lot of energy, but in the rest of their body, they still have things going on. This sodium-potassium pump is one of those things. And even when someone dies, those will continue pumping until the ATP is run out. Because this is a chemical function. It's interesting, kind of morbid, but interesting to go into what happens upon death of organisms. One of the things for mammals is that the sodium-potassium pump keeps running until it runs out of easy-to-find ATP, then it stops. Well, then those start to leak across. In our muscle cells, we have calcium ions, remember, that are stored up in that smooth ER. Same thing, there are pumps that keep pumping them in there. Those pumps also continue for a little bit, up to 4 to 8 hours, depending upon the temperature. Again, they'll keep pumping the calcium ions into the smooth ER until there's not enough ATP. Once the ATP is gone and the pumps turn off, the calcium starts to leak out, and that causes muscle contraction. What's that called? When someone dies and their muscle contracts, all of their muscles. They become really stiff and rigid. Nobody knows. Somebody does, you guys just aren't saying. Rigor mortis, ring a bell? No? Rigor mortis, you guys probably have seen enough shows to know that mammals, not just a person, but mammals get stiff a few hours after death. That's why. And then they continue that. So the calcium ions are leaked out and the muscles will continue to contract until their supply of ATP runs out. And then once they run out, then it'll relax. and the body becomes movable again. That's part of forensic science, where if you went into that, you would study like that chemical process and how long does it take for those conditions, how long does it take at this temperature, how long does it take at this temperature, because it's very temperature dependent. When it's colder, it's slower. When it's warmer, it's faster. And so when somebody goes to, say, a crime scene and they're trying to figure out the time of death, one of the things they look at is, are they in rigor mortis? Because then that at least gives them a window, because they know that it starts from 48 hours and it goes, I don't know, like 24 to 48 hours or something like that. So that tells them at least it's that recent. Then they're going to adjust for what temperature it is. If it's really cold, that means that it could be longer since they died. If it's really warm, that means that it's been hours, hasn't been very long. So another thing they might do is take the temperature of the body. If it's higher than the surroundings, it hasn't cooled off yet. So that means it's been more recent. If it is the same temperature as the surroundings, well, then that doesn't give them a lot of information. It's been long enough that it's reached that temperature. So that kind of stuff is kind of interesting. There are other ways that I also think are gross, but smart ways to assess how long it might have been. So if the body's been past rigor mortis, then that they know it's been at least this long, but they may not know how much longer. So of course they can look at extent of decay, but if not a lot of decay has started, then they have to start looking for other signs to try and backtrack and figure out when they died. One of the things that they can do is it's called a forensic entomologist. They study bugs, insects. And so they will call somebody like that in to see if there are any insects that have laid eggs on the dead tissue, because that's what these things do. And they can smell it sometimes from miles away, way before we can smell anything. And so they will come immediately to that site, usually within like 24 hours, some of them, and start laying eggs. And so this scientist will examine the body for various types of eggs, or maggots, flies, other insects. They each have a different breeding cycle. Some is longer, some is shorter. And based on where they're at in that breeding cycle, that'll tell them how long it's been since they were there and laid eggs. And so it's been at least that amount of time since they died. And depending on what species it is, if they can start smelling that within, say, 2 hours, then that will give them a much better time block to figure out when they died. So that kind of stuff is interesting. All right. So sodium-potassium pump, constantly going, that's active transport. It's not the only type of pump. Like I said, we also have calcium pumps and some other things. If you guys take A&P, you'll spend a lot of time talking about the sodium-potassium pump. And it's interesting how it works. These are both positive ions, but in our bodies, there's a lot more sodium than there is potassium. So if you compare the numbers inside the cell versus outside, potassium's inside, sodium's outside. So sodium is more positive than the potassium total number. So outside's more positive compared to the outside, then the inside of the cell is negative. So that you look at the whole charges, and then you look to see what happens, say, when a nerve impulse fires, because that's an electrical charge. So how does our body generate an electrical charge? This is how. So the reason why the body does this is to, A, set up some ability to transmit some sort of message by an electrical charge, and B, anytime you have a concentration gradient, that is a huge source of potential energy. And so we'll see this again, not sodium potassium, but a different type of molecule in the mitochondria. When ATP is made, the energy comes from creating a concentration gradient. So here what happens is, along an axon, once it starts to generate an impulse, gates open, and it allows the sodium to flood inside the cell. So inside was negative, now it's not, because you had all those positive ions come in. So now it's more positive than outside. So it flip-flops that charge. So we had negative, positive. It swaps places, and that generates the transmission of a charge that way. because it'll do that and go all the way down the axon that way, with all of the sodium bridges opening. And then right after that, the potassium bridge is open. So initially, the potassium is still stuck inside, even though the sodium's flooding in. But secondary, right after that, the potassium doorways or bridges open, and they go back out. And so then you reestablish what the normal charges were. Then after it's completely passed, the bridges close and the pumps are still pumping, and eventually it gets things back to where they should be. So after it fires, initially there's what's called a latent phase. That axon, that nerve cell, cannot fire another signal for a certain amount of time because it has to reestablish that concentration gradient. Muscle cells are the same way, where if you work them to exhaustion, part of what you've done is, if you've sustained contraction for long enough, if all the calcium ions are out and the ATP runs out, there's going to be more as your cell makes it, but if you are working so hard that you exhaust that supply before it's remade, then that cell has a latency period, that muscle cell. It cannot contract until all those calcium ions go back into the smooth ER and the ATP supply is replenished. So because of that, when muscle cells contract, when a group contracts, it doesn't use all of the fibers at one time unless it has to. Now, the harder you're working, the heavier the weight or whatever it is that you're doing, the more muscle fibers will be recruited. And so you can get to the point where you would exhaust, say, the whole bicep. That's unusual, but it could happen. All of that's just, I find, obviously pretty interesting. Coupled transport. Coupled transport is when two things move in or out together. When you have two things move in or out together. Okay. They give you an example down here. Sugars and amino acids. They are separate organic groups, but they tend to pair up. Okay, so it says coupled transport. Those sugars and amino acids usually pair up and come into the cell together. Sometimes these things will couple, but it's not necessarily what the cell is trying to do, like sodium. So I said sodium is supposed to be pumped out, but it constantly finds a way to sneak back in. The sodium ion is really small, and so it can easily hide itself within another molecule. And it does that with these sugars. So it can find a spot on glucose to get in and kind of stick. And our cell, of course, is constantly pumping in glucose. So it's kind of like smuggling across a border. So that's what's happening with the sodium. It will constantly do that. So it hides in the glucose and gets in that way. Potassium can do the same thing, getting out, where it can hitch a ride on another molecule and get out that way. This just shows the rates of exchange. At some point, it's going to taper off because for any pump or for any other protein that might be facilitating diffusion, if they're all busy, that's when it's going to taper off. So here what they were looking at is how fast is the uptake for glucose. Well, it's going to increase as the concentration outside increases. So the more glucose is outside the cell, the faster it'll uptake to a point. So up here we see that it's going to taper off because there are only so many proteins available to bring that glucose in. And if they're all busy, it's kind of like sandbagging. If you have people sandbagging, even if you have the fastest people, it's going to taper off at some point where you can't move any faster because you only have so many people. Your cells only have so many proteins, and once they're all busy, they're all busy. All right, so the last thing we have to cover is this big stuff back here that I skipped over. Endo and exocytosis. Okay, so this talks about bulk passage. Bulk means big, either big pieces or big quantities. And they separate it either into the cell or out. Endocytosis means coming in. Endocytosis, that's bringing stuff into the cell. Exocytosis then is out of the cell. I'm going to give you 3 types of endocytosis, but only 1 type of exocytosis. Here's 2 of the endocytosis forms. Phagocytosis is like, they usually say it's like cellular eating because the particles are solid. So in phagocytosis, it's a solid piece or pieces that are coming in. This is showing a white blood cell engulfing a bacterium. And the size difference really is that big. Our white blood cells are huge compared to bacteria. And so it finds one, it engulfs it this way, and brings it in a vesicle. What organelle will digest this? Lysosome, good. When the lysosome digests this, if some of the parts are toxic, where does that go? What organelle will detox? Smooth ER, good. And then if there happens to be extra, so when it does that, it keeps any usable parts. So for proteins, it'll break them down in amino acids, and if that cell can use those amino acids, it keeps it. It keeps anything that's usable. And then what it considers junk is going to leave the cell, typically by exocytosis. We'll see that in a minute. Pinocytosis. This is like cellular drinking. Here, it's just a bunch of liquid. So the fluid outside the cell. This happens sometimes if the cell is growing rapidly. It needs to increase its volume quickly. It will just engulf a bunch of fluid outside and bring it in. Or if the cell is in great need of fluid, say if you're dehydrated and you start drinking a bunch of water, so all of a sudden there's excess water outside the cell and engulfs it and brings it in. Since both of these change the shape of the membrane and then bring a vesicle in, the vesicle's moving around, all of that takes ATP. So these are also a form of active transport in the sense that they use energy. So endo and exocytosis could go under here. Some books will tell you that they don't include those for active transport because they only want to include the protein pumps and what's happening at the membrane. I prefer to include it just because if we're separating things based on their energy use, I think it makes more sense to put them here. It's more logical to put all of it, anything that's active, in one group. So anytime we're talking about changing the shape of a cell, that's going to use energy. So these are going to use energy. The third type of pinocytosis has a long name. It is called, give me a second, I'm having a brain fart. Thank you. Receptor mediated endocytosis. Receptor mediated endocytosis. This form of endocytosis is very specific. That's what makes it different. Very, very specific. These two are not. Phagocytosis, anything solid. Pinocytosis, anything liquid. Receptor-mediated endocytosis, it's looking for one type of molecule. So if there's a certain type of cell that needs to accumulate some amino acid that other cells in your body won't use, then it will gather it by receptor-mediated endocytosis. Okay? Your liver takes the excess cholesterol out of your blood. This is how it gets it, by receptor-mediated endocytosis. The receptor here that they're talking about is a protein. So it's a protein that's on the surface of the cell membrane, and it has, remember we looked at protein that maybe had some sort of shape like this. in the cell membrane. And out here it showed that, oh, that's the molecule that fits it. Okay, so that's what this is. Only this molecule can hook up to this protein, and when it does, that protein pumps it in. Any other molecule that has some other shape, it can't bind to this, and so then it's bypassed. Exocytosis, there's only one kind. It's just when a vesicle is full of something. Could be solids, could be liquids. It moves to the cell membrane and dumps out. Now, this could be a secretory product if that cell is secreting something. Okay, so mammary glands in cows and other mammals, humans, they secrete milk like this. So the milk is made inside those cells, and then it moves to the membrane and dumps out. And then it dumps out into channels. Eventually, they all come together. If you look at a cow's udder or something like that, there's a bunch of them around the nipple. But they come from, a whole mass of cells that are doing this. Any cell that makes hormones does it this way. But it doesn't always have to be some secretory product that's like beneficial. It'll do this with waste. So from that bacterium, if a white blood cell digests the bacterium, keeps all the good parts, it may have a bunch of stuff that is waste. It does this with it. And here's what it actually looks like, where we see some solid products coming out of this cell. Or this vesicle, I should say. Questions over that? Okay. Chapter 8. Okay, so the test is going to be over chapters 4, 8, and 9. 8 and 9 go fairly quick. Okay, so chapter 4 was all about cell organelles and cell transport. Chapter 8 is over mitosis. Chapter 9 is over meiosis, both of which are different types of cell division. So the first thing they hit are the differences between prokaryotes and eukaryotes. Prokaryotes have a simple cell cycle. What's a cell cycle? A cell cycle is from one division to the next. Well, prokaryotes are always single celled, and remember there's no organelles that have membranes on the inside, so they're small. There's only one chromosome if we're talking about bacteria, only a few if we're talking about archaea. So because of all that, their cell cycle is much simpler and can be very fast as like 20 minutes. So that means for certain bacteria, strep, for example, you get strep throat, it can come on really fast because every 20 minutes they're doubling in number. So you pick up a few from wherever. By the next day, that's why you have a raging sore throat, is because every 20 minutes they've been reproducing and doubling their numbers. They give you 2 steps. I'm going to give you 3. The first stage is to copy the DNA. That's called replication, DNA replication. For bacteria, they only have 1 chromosome, so that doesn't take very long. The second step that I want you to add is for the cell to grow bigger or longer. The cell grows bigger or longer. And then make this number 3. It splits. Make number 2 right here, number 3. It splits. So number 1, making an extra copy of DNA. So now there's 2 copies. Number 2, it grows bigger. And number 3, it splits in half so that we have 2 new cells. That process is called binary fission. Fission means to split. Even in chemistry, fission means to split. I mean binary because it's splitting in half or into 2. I'm not going to ask you about this origin of replication, so you can read about it, but I'm not going to ask about it. here's what it looks like. So here's the original cell. It's going to copy the DNA first. This is a bacterial cell. It has one circular chromosome. What they're showing you here, that's where they were getting into the origin of replication, whatever. So it starts at one spot, of course, and it goes out from that until it has 2 full chromosomes. Now, it doesn't really show you, but these are actually attached to one single point on the membrane, so that when it grows longer in that second step, it's moving these 2 apart from each other to make sure that there is no entanglement of the DNA and there's enough cytoplasm for 2 cells. Once there is, then it splits in half. We now have 2 bacteria. These daughter cells, I don't know why it's always a female inference, whoever first came up with these, but the 2 new cells are always called daughter cells. It doesn't matter if they have male or female parts. Most cells don't have one or the other, but some sex cells do. These are identical. In binary fission, these daughter cells are now identical to one another and to the parent cell. Notice that the chromosome is made of one old strand and one new strand because the old strands were purple. The new strands we're seeing are red. We'll cover that again later in another chapter. And so that's pretty easy. That's simple. Eukaryotes, of course, much more complex. Our cells are so much bigger, have so much more DNA and chromosomes, so many organelles, that it takes quite a long time in comparison to get them ready to split. About 24 hours. Now it's going to vary from one cell to the next depending upon how big it is, but usually we'll say an average of 24 hours. The DNA is the first thing that we see that's much different. In eukaryotic cells, there's multiple chromosomes. There's a lot more DNA, and there's a lot bigger chance that it's going to get tangled and broken. So it takes longer to get them ready. It says eukaryotic cells have 2 mechanisms to split the DNA. Nuclear division. So truly mitosis is splitting of the nucleus. It's being kind of picky on that, but it's splitting of the nucleus. Normally, we also get cytokinesis, so what you were viewing today in lab. Occasionally, you'll get a cell that goes through mitosis, but not cytokinesis. Well, you end up with a cell that has more than one nucleus. That happens typically only in a very big cell that needs more than one copy of DNA because it can't keep up with all the proteins that need to be made unless it has more than one copy. Our skeletal muscle cells are that way. Okay, so in your bigger arm muscles, leg muscles, that type of thing, they're so big that typically they need multiple copies of DNA, and they're protein-based, so they're making a lot of proteins, so they need multiple copies. Most of the time, though, mitosis and cytokinesis go together. Now, mitosis occurs in non-reproductive cells. What they mean are, this is not used to form sperm and egg, but everything else. So all of the other cell division that we talk about is mitosis. So, for example, when you get a paper cut and your finger heals, that's mitosis. When you shed skin cells and more is constantly being made from the deepest layers, that's mitosis. Binary fission that we just looked at with prokaryotes, that's mitosis. So almost all of cell division that we talk about is mitosis. Meiosis is for one thing only, and that's to form sperm and egg. And so let me have you guys write that down, because I know I asked about it on the test in the form of short answers. So functions of mitosis. So what is it for? Well, number one, growth. Growth of an organism is what I'm referring to here, where you need more cells. Okay, so the cells grow a little bit. You guys saw that some of the newly divided cells were a little bit smaller than the other ones. So yes, they grow. But when we're talking about growing from a zygote all the way up to an adult human, that would be ridiculous to say that we're all born with the same number of cells and they just get bigger. We grow by mitosis so that there are more cells. Number two, repair. Like that paper cut or any other damage that might have been done. If it has to make new cells, then it will. Now, not all cells have the ability to do that. You think about spinal cord cells, nerve cells, brain cells. We have recently found, we thought that brain cells couldn't reproduce. We've actually found that they can. We just don't know how to instigate it. So once we could figure that out, that would be awesome because we could fix a lot of things. For other organisms, repair might be like starfish. If you cut off one leg, they grow another one. If, this is pretty cool, if you cut off one, not that I would torture a starfish like that, but if it loses one leg and part of the main body part, I mean the central part, the one leg with the part of the central body will grow into a whole nother starfish. This one will grow another leg. And so then you end up with two starfish that are identical to one another. That's kind of cool. Do what? No, I would say days, maybe a week or two, but it happens pretty quickly. It's kind of, I mean, since that's a eukaryote, how fast our skin heals would be kind of the same. Now, it's not just their epithelial tissue, it's others too, but pretty quick. And then finally, the third one is asexual reproduction. Asexual. Asexual reproduction means there's only one parent. In asexual reproduction, there's only one parent, like binary fission. Now, this isn't just for prokaryotes. Some eukaryotes can do this too. Yeast cells, for example, yeast is a eukaryote, but they can divide by budding. So in budding, you'll have this yeast cell that starts to just grow a little growth off to the side. And that'll grow into a whole nother yeast cell and they'll pop apart. All right, somatic cells. These are the cells that divide by mitosis. These are considered typical body cells. Somatic cells. They divide by mitosis. For us, it's a typical body cell. For any other mammal and most other animals, also just typical body cells. Even plants, you could say that. Almost all of our cells are somatic cells. The only cells that are not are sperm and egg and the parents of those. The cell that splits into sperm and egg, those are called germ cells. Germ cells divide by meiosis. Germ cells divide by meiosis. Where does the term germ come from? It has a different meaning here than it does in our typical language. When we typically talk about germs, we're talking about microbes that make us sick. This means anything that is a germ-line type of cell in biology means it has the ability to divide. So these germ cells divide into either sperm or egg. Mitosis, a little bit more about that DNA. In mitosis, the two daughter cells are identical. Should be. They're identical. The number of chromosomes is the same as the parent cell. So one of the questions in your lab book, one of the last ones today, was something about parent cell has 16 chromosomes, divides by mitosis. How many do the daughter cells have? 16. Okay? So 16, because it's mitosis. Now, in meiosis, those daughter cells have half the number. If you think about why that would be, humans have 46 chromosomes in all of our somatic cells, even in our germ cells, but our sperm and egg have 23. We have to go through meiosis so that a baby has the correct number of chromosomes. Other animals and mammals, same thing. If they don't half the number, they don't split the number of chromosomes before fusion of sperm and egg, then the resulting zygote does not have the correct number. For us and most other mammals, that would result in miscarriage. Now there is one mammal, it's the only one we've ever found, that has twice that number, that came about, evolved because meiosis didn't take place and the sperm and egg cells fused. So it's a little, it's a type of a rodent, rat I think, that's down in either South or Central America. It has 4 sets of chromosomes in all of its somatic cells. We have 2. We get 1 set from mom, 1 set from dad. In that little rat, something happened, who knows how long ago, that meiosis didn't take place, the cells fused, and so then it ended up with 4 sets of chromosomes. It's very unusual because most of the time, even if we have 1 extra chromosome, our zygotes don't develop. It ends up in miscarriage. So how that happened with some other mammal that ended up with 4 sets is pretty special. All right, cell cycle, from one division to the next, remember. From one division to the next. For eukaryotes, we have to divide this up into interphase. Interphase is the time between divisions. And then what they call the M phase and C phase, I have never called it that, but mitosis, or the M phase, cytokinesis, UC. These two go together because almost always cytokinesis happens with mitosis. Only those relatively few times does it not happen and we have a cell that has more than 1 nucleus. All the other time, so when it's not dividing, all of the other time is in interphase. Okay, interphase, when they first started studying this, they were studying, looking at live cells as they were dividing and using a light microscope. And what they saw was that the time between divisions, so you can imagine looking at the various stages of PNAT like you did today. In a live cell, you'd be able to watch it go through those stages, and then it goes back to 2 daughter cells that each have then the nucleus and the nucleolus. And it might get a little bit bigger, but other than that, you're not seeing anything happen. And so when they were first looking at that and watching it occur, they named it interphase because they thought it's a resting phase, it's just in between divisions, and didn't think really anything was going on. Well, we know now that there's lots of stuff going on, and we can divide interphase into 3 parts, and that is G1S and G2. Now, interphase is really long compared to mitosis. Interphase is probably 90% of the time is spent in interphase. And that's for cells that continue to divide, like your skin cells. They're constantly dividing. Our liver can repair itself. So if it gets damaged, it can divide to repair itself. That's why in extreme cases, you could donate part of your liver to, say, a family member if they couldn't find a matching donor, because your liver will grow back and the part you give them will grow. So that's kind of neat. All right, so interphase. The first phase of interphase is called G1. I remember it as growth 1 Okay, growth 1. Now, really what happened? Kind of the same thing, where, okay, they started to really study this interphase more, and they saw that, you know, at the beginning it seems like it's not doing anything. Well, so they called it gap, because it was a gap in activity. That's what G actually stands for. I don't remember it that way, because now we know that's not the case. So this is primary growth. This is when you start with a new little baby cell and it gets bigger so that it becomes full adult size. That's growth 1. It says most cells spend the majority of their lifespan in this phase. For any cell that does not seem to have the ability to reproduce anymore, this is it. They are reproduced, they hit G1 and they stay there. So for example, our brain cells or spinal cord cells, they're living, but they never pass this G1 phase, or we haven't figured out how to induce them to. When does that happen? For some cells, like our skin cells, that's just constant. It just keeps going through all of this. For your liver cells, they stay in G1 unless something happens where they're damaged and they can sense that there's damaged cells nearby. Then that will spur them on to go into the S, the G2, and then through mitosis to reproduce. But otherwise, they're just sitting in G1. Okay, G1, longest of the 3. G1 takes probably more time than the S phase and G2 together. So this is the longest part. All right, after that, the S phase. Only cells that are going to divide go past G1. So S and G2, that's only for cells that are destined to divide. I mean, at that point, if they hit S and they go through it, they're heading for mitosis. S is DNA replication. The S stands for synthesis because it's synthesis of DNA. So DNA replication. During G1, all of your chromosomes are single-stranded. Because remember upstairs when we were just looking at PMAT, they were all double-stranded, then they get pulled apart. Once they're pulled apart, they're single-stranded. So in telophase, and then back going into interphase in G1, they're all single-stranded. So we have 46 that are single-stranded. The S phase makes duplicates. That's when they become double-stranded again. So that's usually how they're shown. When people think of chromosomes, they always think of the X-shaped chromosomes. But the truth is, far more often, they're single-stranded, not double. The only time they're double-stranded is at the end of S, then G2, and mitosis. And so here, all chromosomes are single-stranded. After S, they're now double-stranded. G2, this is growth 2. The growth that happens here is specific for mitosis. So at this point, the cell's really close to dividing the nucleus and possibly then also cytokinesis. They have to make sure that there's enough organelles for 2 cells. All the microtubules have to be grown to help split those chromosomes. For animal cells, the centrioles, that's when they're made, is here in G2. For plant cells, the aster comes about in G2. Once this is done, then it heads right for mitosis, which they're calling the M phase. Mitosis is pretty quick. Once it gets here, it's going to proceed through those 4 phases pretty quickly. And a lot of that is because how the DNA exists. It is now chromosomes. If we were to look back at interphase and S and even G2, so G1, S, G2, all during interphase, there's no chromosomes. It exists as chromatin, even though I said chromosomes are single-stranded, not double in, say, G1. But they don't actually look like those chromosomes that we typically see because it's all unwound. Because during that time, that's what the cell is doing what it's meant to do. So if it's producing hormones, that's during interphase. If it's producing a lot of energy, that's during interphase. So whatever the cell is supposed to do, that's what it's doing during interphase. During mitosis, that's just for dividing. And because the DNA is wound up in chromosomes, it's like that skein of yarn. If that cell needs a protein that is specified by DNA that's on the very interior part of that skein of yarn, it's not happening because it's not accessible at that point. So that's part of why this is happening so quickly, is because when the DNA, when the chromatin winds up to form chromosomes and it condenses, most of it's not accessible. It can't be used. And so that means there are no proteins being made that typically are. So it has to be pretty quick. All right. So on Wednesday, we're going to finish mitosis and probably start chapter 9. So make sure you come with your chapter 9 PowerPoints. I'll see you then.